Learning outcomes. After studying this module, you shall be able to know about various tests performed for semen analysis. You will learn about the techniques for examination of semen. You will be able to identify unknown fluids found at crime scene. You will also be able to analyze various types of sexual assault cases. Introduction. The ability to identify traces of body fluid discovered at crime scene is very important aspect of forensic investigation. Semen analysis is one of the most popular and informative forensic techniques. It is imperative that any potential body fluid sample is properly collected and not destroyed during the initial identification process. They can also help in answer to various questions regarding the events of a crime. An analytical technique that could identify a particular body fluid rapidly, simply and non-destructively at the scene of a crime would be a valuable tool for forensic investigators. In addition to blood, semen is one of the most prevalent body fluid found during criminal investigations, especially in cases involving sexual assault. There are currently several tests, both presumptive and confirmatory, that can be used to identify an unknown fluid found at a crime scene to be semen. An alternative light source can be used as finding aid to detect certain biological materials including semen. Some commercial ALS instruments, that is alternate light sources, have been developed such as the Woodslam Bluemax. Seminal stain is an important biological fluid which requires identification and characterization in the investigation of sexual offences like rape, sodomy, bestiality, etc. Normal semen reference pH range is 7.2 to 7.8. Variations in volume and low pH may be due to congenital abnormalities of the genital tract or to the obstruction. A pH of 8.0 or greater can be associated with infection of prostate, seminal vesicle or epididymis. Now let us learn about the examination of semen and seminal stain. First we will do the visual and physical examination in which we try to check the color. The color is usually thick, yellowish white, glary, secretion having a characteristic odor known as seminal odor. Texture. On touch, seminal stains are stretchy. Appearance. Garments sent for forensic examination are usually dirty, having variety of stains. In natural light, some stains are reddish colored, while others are brown, yellow or faint grey in color. These are often mixed with stains of blood, vaginal discharge, urine and semen. So, as to restrict the investigation to seminal stain only, preliminary examination is done under filtrated ultraviolet light. Next step is to do microscopical examination. Microscopically, detection of spermatozoa is also considered as confirmatory evidence for the presence of semen in a suspected stain. In order to conduct microscopic identification, part of the stained garment should be dissolved in acidulated water in a test tube. The test tube should be subjected to ultrasonic oscillation, which can separate the spermatozoa from the dried stain, followed by microscopic examination. After fixing the same in dilute sulfuric acid, since spermatozoa contain gram-positive protein, the presence of spermatozoa will be confirmed by microscopic appearance of purple bodies. Next we move to the presumptive test. First is acid phosphate test. The acid phosphate test is one of the best known and most widely employed technique for the purpose of identification of semen. The test is adopted because of high acid phosphatase activity of semen. Thus, by applying the test, a suspected azoospermic or vascuitomized person who is a suspect of sexual offense may be identified. Stability of acid phosphatase activity in seminal stain depends on the temperature. Suppose if a stain is exposed to sunlight, the acid phosphatase activity is slowly reduced, and if the stain is heated at 60 degrees Celsius for 5 minutes, this activity is completely destroyed. The enzyme acid phosphatase of semen hydrolyzes the calcium alpha naphthyl phosphoric acid that liberates alpha naphthol. This couples with the H-forming material to give purple color. Now let us see the reagents preparation. First is the buffer. We use glacial acetic acid 1 ml, sodium acetate anhydrous, sodium acetate anhydrous 2 gram, distilled water 100 ml. Now step 1 is to prepare reagent. We take buffer 50 ml and then sodium alpha naphthyl phosphate 0.25% 126 gram. Step 2 reagent, buffer 50 ml, naphthanyl diazoblue B 0.5, that is 250 gram. Now step 1 reagent and step 2 reagent can be made up in bulk and aliquot into test tube and frozen. When needed, 1 tube reagent can be put under the warm running water for use. Now let us see the procedure. Place a small piece, that is 2 into 2 mm of suspected seminal stain. Stain material on Whatman filter paper on other suitable test paper. Use proper standards and controls including positive, negative and unsustained controls. Add 1-2 drops of step 1 reagent and allow to react for 30 seconds. No color should be developed at this stage. Then add 1 drop of step 2 reagent and record the result after 10 seconds. A positive reaction is recorded upon rapid development of a semen. This is a confirmatory test for semen. Next is thin layer chromatography. This method is used for detection of colon and spermine. In TLC, it is possible to detect colon and spermine simultaneously in seminal stain. It has been possible to detect colon and spermine in old seminal stain events after the lapse of 5 years by this method. There are 2 types of reagent used in TLC method. They are Dragonroff reagent and potassium iodoplatinoate reagent. Basically, TLC works on the principle of separation according to their affinity towards the relative phases. Next is examination through ultraviolet light. 11 commercially available pieces of clothing of different fabric types were tested. 3 to 100% cotton, 3 to 100% polyamide, 1 to 100% polyester, 1 to 95% cotton, 5% spandex, 1 to 73% cotton, 24% polyamide, 1 to 3% spandex, 1 to 80% polyamide or 20% spandex. This resulted in different colors and color combination of fabric, including white and black. Now, the fabric was classified into three groups: bright, that is the white, bright blue and pink; medium, blue, green, yellow, orange, red; and the dark, that is the black or brown. Now next is the light. To test the excitation of the samples, we used a newly developed very high intensity light source manufactured by Lumatic, Germany. The portable system is suitable for heavy use at crime scene because of its low weight, that is approximately 6 kg, an unusually robust full metal shell, compactness, and a battery that allows 60 minutes of operation away from electrical outlet. A mercury lamp inside of a unit produces a high intensity, that is around 320 to 400 nanometer, and visible light, that is 400 to 700 nanometer, which was developed to detect biological stains even in daylight. And at the end of the flexible light tube result in a beam applicator focused on a spot or spread to fan-like triangle. Wavelength can be chosen by 11 adjustable filters position, that is 400 to 700, 350 to 500, 400 to 500, 320, 400, 415, 440, 460, 490, 550 and 570. In combination with the 11 excitation settings, white, that is below 400 nanometer, orange, that is below 500 nanometer, and red below 590 nanometer were used to block the excitation light and to visualize the fluorescence more precisely. Next we have the confirmatory test, that is the chemical test. Crossover electrophoresis. Seminal material can be identified by demonstrating the process of P30, a semen specific protein. One method of doing this is crossover electrophoresis. An extract of suspected stain is placed in cathode well of a gel plate and anti-P30 is placed in the anode well. Electrophoresis is commenced for 20 minutes at 200 volts, forcing two components together. When the antigen, that is P30, meets the antibody, a precipitin band is formed. The presence of precipitin band within an extract of unknown stain proves that stain contains seminal stain, that is anti-P30. Next is plate preparation. Clean 1:3 or 2:3 microscope slides with detergent, resin and polish dry. With a diamond tipped scribe, number the slide in the upper right hand corner. Slide should be consecutively placed or numbered. Next is pre-coating plates. Note, omit this step if gel bond is used instead of glass slide. In order for final 1% agarose to adhere to the glass slide, it is necessary to pre-coat the slide with thin layer of 0.2% agarose. Then add 1 gram of 0.2% stock agarose to the 10 ml of distilled water and boil until dissolved. Paint a thin coating of this solution onto the pre-numbered glass slides with a soft brush. Wipe the edges of the slides with tissue paper or chem wipe to remove any overflow. Allow the air dry. Then store pre-coated slides in a numbered slide box for later use. They can be kept indefinitely. Next is coating plate with gel. On a level surface, place a pre-coated slide onto a small inverted beaker, allowing the slide edges to be suspended. Place 1 or 2 tubes containing 3.5 ml of 1% agarose gel in a water bath to liquidify. Then 3.5 ml tube for 1:3 slide, 3.5 ml tube for 2:3 slide. Pour the liquefied 1% agarose onto the slide. Surface tension and pre-coating of slide will prevent the liquid from overflowing the edges. A smooth level surface is formed. Allow it to cool and harden. Now next is fluorescence test. This test was discovered by Dr. Florence in the year 1886 when fluorescence reagent, that is potassium iodide, iodine plus water, is applied to the slide. It produces rhombical shaped dark crystals of colon per iodide. Similarly, any tissue or biological material containing sufficient high colon concentration would give positive fluorescence test. According to Davis and Wilson, if swab is collected within 1 day of sexual act, colon can be detected. At the same time, if swab is taken after 14 hours of intercourse, there is possibility of negative test. Next we have Barberio test. This was invented by Barberio in year 1905 when the question stain is allowed to react with picric acid. It leads to the formation of yellow needle shaped spermine picrate crystals, including the presence of seminal stain. Next is detection of seminal stain. Sandsberg in 1977 reported the event of isolation of P30 in human seminal fluid. It is a glycoprotein of prostate origin. Human seminal stain of up to 1 year old gave positive result for P30. Vaginal washing collected from the victim of sexual assault will give negative result for P30. Detection of seminal stain is placed on the crossover electrophoresis. Suspected stain extract is placed on the cathode and P30 is placed on anode and electrophoresis is conducted for 20 minutes at 200 volt, leading to the formation of precipitin band, confirming the presence of P30. Next is the morphology of human spermatozoa. The maturation of cells in seminiferous tubules to mature oval spermatozoa with an acrozonal cap in the ejaculated involve differentiating steps. Some cells will prematurely terminate the process or result in other shapes, tapering heads, pinheads, round heads. It is important to note that abnormally shaped sperm usually do not participate in fertilization, but if fertilization occurred, there is no evidence to support an increased risk of malformation of developing fetus. A semen sample usually contains 50% or more normal spermatozoa. Items that have been tested presumptively positive for seminal fluid using AP test can be confirmed either by microscopic detection of spermatozoa or chemical detection of semen specific protein. Positive swabs or a small cutting from a positive stain can be simmered onto the microscopic slide and then stained for visualization. Two common stains used for visualization of spermatozoa are nuclear fast red, that is red stain, and picoendocarmine, that is green test, and sometimes referred to as Christmas tree stain because of the red-green color combination. Once stained epithelial cells, that is a group of cells such as skin cells and cells of the lining body, orifices and spermatozoa have a specific appearance. Now microscopically, identifying spermatozoa is an absolute indicator that semen is present on an item. It is also useful because the relative quantity of spermatozoa and epithelial cells can be assessed. This determination becomes important during subsequent DNA analysis because spermatozoa contain male DNA while most epithelial cells in the male-female sexual assault will contain female DNA from complainant. Now let us see about protein conformation of semen. Traditional P30 detection test utilizes electrophoresis or diffusion method such as crossover electrophoresis and Autrillon's double diffusion or ELISA. Commercial test kits for P30 are also available and have become prevalent in forensic laboratories because of their sensitivity and ease of use. All these methods required a small cutting of an AP positive stain to be incubated in water or saline until rehydrated. Afterwards, the liquid is separated from the cutting by centrifugation so that the stain will be retained in a liquid form instead of dried to be cutting. At this point, a portion of extracted stain can be used to test for the presence of P30. All of the methods that detect P30 rely on the formation of an antibody-antigen complex. If semen is present in stain, the binding of P30, the test antibody produces a visual result. The lack of a result in any of the tests would indicate that the stain does not contain semen or that not enough to present to facilitate a visible reaction. This test can be used alone to confirm the presence of semen, or it can be used in relation with microscopic method described in the previous section. Now let us see the application of forensic in semen examination. Semen analysis is an important laboratory test and should be thought of in the same way as any other diagnostic is used in determining treatment plans for infertility. Semen analysis is very important in assault cases as it is very useful for detection of semen on the clothes. The isolation and identification of seminal stain found on various suspected areas such as cloth, bedsheet, pillow, blanket, etc. Analysis of various proteins present in the semen or seminal stain. The seminal stain examined generally in rape cases, sodomy, bestiality, buccal coitus and cases of sexual perversions. detection of sperm cell in vagina and other body parts, fecal matter examination. Now let us summarize what we have learned in this module. Through the various techniques used in semen analysis, it can be examined that the stain found at the suspected area is semen by analyzing the spermatozoa presence. The spermatozoa found in case of women is generally spindle shaped, having a motile tail and head in which components such as P30, choline, spermine, amino acid and some proteins are found. A very fine and clear structure of spermatozoa can be seen by the help of compound microscope. The presence of choline can be detected by using enzymes such as choline ester of semen or seminal stain. Thank you.